Hi, everyone. My name's Qaiser. Today, on behalf of all my fellow co-authors, I'm going to briefly walk you through our new manuscript, which is titled Genome Sequencing Identifies Rare Tandem Repeat Expansions and Copy Number Variants in Lennox-Gastaut Syndrome. This is now published in Brain Communications. As you may know, epilepsies are a group of common neurological disorders which are primarily characterized by recurrent seizures. We know that genetic factors contribute to epilepsy, where to date, over 900 epilepsy-associated genes have been identified. But despite this known genetic basis, the molecular diagnosis of epilepsies remains challenging, with current studies only capturing a portion of the contributing genetic variation. There are many reasons which can explain why, including the incomplete assessment of genetic variants by most current genetic analysis methods. By looking across the entire genome using technologies like whole genome sequencing, we can explore new insights into epilepsy genetics and help resolve epilepsies of unknown etiology. And that's what we did in our new study. We recruited a cohort of 30 adults with unexplained developmental and epileptic encephalopathies, for whom prior genetic tests were inconclusive. In total, our patients have completed 55 molecular genetic tests, including large gene panels or whole exome sequencing. Using whole genome sequencing, we explored different types of rare genetic variation. If you've never heard about genetic variants before, let's do a little thought experiment together. Picture your DNA, your entire genetic code, as a book. Similar to how letters make up words in a sentence, bases in our DNA form genes. Using DNA sequencing, we can go through all the chapters in your book and search for different types of changes, which are called genetic variants. Sometimes these changes can include single typos, which are called single nucleotide variants. There might also be torn out pages, like a deletion, or duplicated pages. These are called copy number variants. You can also find a long series of repeated words. These are called tandem repeat expansions. Using whole genome sequencing, we can search for these different changes. Every time we find a change, we evaluate the genetic variant using guidelines from the American College of Medical Genetics and Genomics. Is the variant pathogenic? Is it likely to cause a disease? Or is it benign? As a result, we're able to find the molecular answers to unresolved epilepsies. And so, using whole genome sequencing, we identified pathogenic or likely pathogenic genetic variants in 32.1% of our cohort, as well as 2 tandem repeat expansions. You can find details about each genetic variant in our full manuscript. Interestingly, to the best of our knowledge, this is the first report of a coding and a non-coding tandem repeat expansion identified in patients with Lennox-Gastaut syndrome. Overall, our study demonstrates that using whole genome sequencing, the examination of multiple types of rare genetic variation, including those found in the non-coding region of the genome, can help resolve unexplained epilepsies. This study would not be possible without the 14 different authors. This includes graduate students, lab managers, postdoctoral fellows, research associates, professors, and physicians. We would also like to acknowledge the funding support we've received from various institutions, and to thank all the patients and their families for participating in the study. And thank you for listening.